Hi there, welcome back to the formulaic approach. Now in this video we are going to be covering the topic of pollination in plants and development of new seed. So this whole thing together covers the reproduction in plants, the reproductive process in plants. Just as you know in animals, reproduction is not complete without birthing, okay, without the organism being given birth to. Then also reproduction in plants is not complete until there is a new seed that has developed. Do you understand? So what happens is this: pollination takes place first. You should know that they take place in angiosperms. Okay, angiosperms are flower-bearing plants, so we call them flowering plants. So those are flowering plants, angiosperms. They are the one that gets pollinated. Now, what is pollination? Pollination is actually the transfer of pollen grains from the anther to the stigma. So whether it is of the same flower or of a different flower. So now to understand pollination, you must first understand the way a flower pretty much looks like. So a flower looks like this. Now let's start from the top here. So this place here is the style of the flower, the style. This place here is the stigma of the flower. This is stigma, okay? It possesses sticky substance that allows pollen grains to stay on it. All right, and there is another side here. Again. So this is the stalk of the flower or the pedicel or the peduncle, okay, or stalk. This is the part that attaches the flower to the stem. So the stem of the tree is here, the branch of the tree is here. Exactly. Now inside, now this place here is called the ovary. All right. Now are called these three together are called the pistil, and they make up the reproductive structure of the flower. This is the female reproductive structure of the flower. This part is the female reproductive structure. Now, what does the ovary do? The ovary produces ovules. It produces ovules, okay, and those ovules produce ovules like this. Ovules are the they contain the egg, okay, the egg cell. They contain the egg of the flower. The female, since it's the female part of the flower, so it's going to contain the eggs, isn't it? All right. I'm going to expand the structures. So these are ovules here. They are ovules. And the arrangement of the ovules inside the ovary, the way it is arranged, is known as placentation. Placentation. Now, why is the name like this? You know, just like we did in reproductive system of human, we talked about the fact that the placenta is the attachment of the is the connection between the father, the mother, and the child. So the mother uses the umbilical cord to connect to the placenta, which then is attached to the wall of the ovary of the uterus of the woman. The same way, there is a placenta here on which the placenta is on the wall of the ovary, okay, such that the ovules which bear the eggs are now attached to the ovules are now attached to the ovules are now attached to the wall like that through that placenta, and that process is known as placentation. There are different types of placentation. We have the axial placentation. We're going to cover that in a short video. All right, so that's the point. Now, then there are now structures that emerge like this. All right, so just on the tip of those structures, you have the anther. What is called the anther? So this is called the anther. It's called the anther. Now this thing that bear them up, this filamentous line, okay, filamentous structure is what you call the filament, okay. It's called the filaments, like you know what, because it's even filamentous, isn't it? All right. So the point of attachment of that anther to the filament is now called the connective. Just somewhere around here, you have the connective, and these together, these three together, they make up the male reproductive structure, which is called the stamen. Okay, the stamen, and the group, this group like this, you know, this is filament and the filament and the filament and the. So this group together, all together, make up the male reproductive structure, which is called the androecium. So in the flower, there are what we call the essential parts and the non-essential parts. So in the flower, we have the essential part and the non-essential part. The non-essential. Okay? I'm still going to bring it up. The essential part is the female, or is the reproductive part. Of course, you should be able to tell that. Now, inside the anther are pollen grains. Okay, the anther houses pollens, and those pollens help in the propagation in the transfer of the sperm to the egg of. Yeah, you're welcome. The egg of the ovules that are produced in here. All right. Now the point of attachment of all these guys is called the receptacle. The receptacle. And this one you are seeing here is the sepal. The sepal. Now the sepal has a function when the flower is mature, and it has only one function. And that function is that it is the site for food production for the flower. It photosynthesizes. That's why it's usually greenish in color, because it's embedded with chlorophyll. It has chlorophyll. Now, but this sepal was not, the primitive function of the early function of the sepal is actually not production of food. It is actually the protection of the flower when the flower is in the bud stage. So you see the flower in the bud stage look something like this. You see it has not opened up here, okay? It is still clear stick here, okay? I don't know, the close up, that is closed up here. So this is the sepal. In doing that stage, this bud stage, the bud stage of the flower, it protects the flower. All right. Now, then I said, now there are now structures here that are like, they are leaf-like, okay? They are leaf-like. But they are not leaves, okay? They are brightly colored. They are brightly colored. And those structures are called, the petals. They are called the petals. They are called the petals. All right. So that is pretty much it. Now, what does the petal do? We've mentioned the function of the sepal. The petal, what it does is, it is the one that, because it is brightly colored, it is one that it serves as an attractant to insects, so that it can help to pollinate this flower. Insects will be attracted to the flower, and they will help in the pollination of this flower. Okay? Or birds will be attracted. Now, why do those birds come? Actually, they're not coming to like the petals. They're actually coming because once they see a brightly colored petal, they suspect the presence of a juice inside them, inside this layer here, that is called the nectar or nectary juice. Okay? Nectary juice. The nectary juice. So that is what they are actually coming to suck out of this flower. Do you understand now? All right, having understood the structure of the flower, let's now see some terms that you have to get acquainted to. Now, I have said that the flower has two parts. They are the essential parts and the non-essential parts. The essential and the non-essential parts. Now, the essential part, I mentioned the female part, reproductive part, which is the pistil, and the female reproductive part, which is the male reproductive part, which is the stamen. Okay, but their collection is called for female. The collection of the female reproductive part, if there are more than one ovary, yes, and their flowers are there has more than one ovary. That collection will be called the gymnoecium. Gymnoecium. The word gene, you already know, refers to female. For example, you hear misogynist, that is if someone who hates women, okay? Or you hear gynecologist, doctor of women. So, stamen is androecium, the collection of those long lines and pollen that you see. They're called androecium. So you can already see andro refers to male. Alright. The other essential parts are petals. Now the petals, you see, are basically sorry. Then the non-essential parts, the petals. Now what the petals do is they basically serve as an attractant. Okay. And a collection of petals is called the corolla. Yes. Then the sepa is another non-essential part. I already told you the function, serving for photosynthesis. And a collection of sepals, is called calyx. Is called the calyx. Now listen, you see petals, corolla, sepal, calyx. Now if we now have the sepals, the petals fused together, there is no space between them, they are not detachable one by one. That kind of petal is called gamopetalous. The word gami, I've already told you, means marriage, together, okay? Then, of course, if it is the sepals that are fused together and not separated, is also gamo, or in this case, sepalous. Gamosepalous. They are written together, it's not a compound word, okay? Gamosepalous and gamopetalous. All right, so with that said, now let's get to the types of pollination. We are going to lead it all the way to development of the new seed. Now, what is pollination? We said it's the transfer of pollen grain from anther to stigma. The first type of pollination is called the autogamy. So you can already see the word auto means self, and I said gamy, the word to come together, marriage, right? Remember the word polygamy, as plenty, plenty, marriage. Make sense? That is marrying more than one wife. Okay. So the point is this: autogamy means self-pollination. That is, the plant will pollinate itself. Now, what happens is there will be the transfer of pollen grain from the anther of one flower to the stigma of the same flower, or the stigma of another flower on the same plant. Is that clear? Now, there are two forms of autogamy. I will explain. There are two forms of autogamy. There is the geitonogamy, and there is the cleistogamy. The geitonogamy, the word geiton is gotten from geitinus, which means neighbor. All right. Now, keep in mind this: that geitonogamy actually is seen from two perspectives. Now, from the perspective of a geneticist's perspective of geitonogamy is that geitonogamy is a form of autogamy. Geitonogamy is a form of autogamy. But an ecologist's perspective of geitonogamy is that geitonogamy is a form of allogamy. Okay? It's a form of allogamy. Why? Because a geneticist will say that the pollen grain is going from the anther of one flower to the stigma of a neighboring flower, which is still on the same branch, the same tree. Okay? The same tree, but there's another flower on it. That will be. So the geneticist is saying that all the flowers are the same. They have the same genetic composition on the same plant. So because of that, the method is called autogamy. Joshua, the method is called autogamy. But to an ecologist, since there is a change in the environment of the pollen grain to another flower, It says it is allogamy. It says it is allogamy. All right, so just take note of those. That's my explanation. All right, now then we now have cleistogamy. Now the word cleistos actually means close. Okay, close. So what happens is that it's cleistogamy is a form of orthogamy that happens in flowers that fails to open up. The flowers does not open up, so because of that, it can only be pollinated by itself. So this one is a true form of orthogamy. Cleistogamy is a true form of orthogamy. All right, I hope that is clear now. Then the second type of pollination is called allogamy. Now, what is allogamy? The word allo means another. Okay, another. So that is, it is the transfer of pollen grain from the anther of one flower to the stigma of another flower essentially. So the other flower is going to be a flower on another plant but of the same species. Keep in mind, pollination can only be possible when the flowers are of the same species. If the plants are not of the same species, they cannot cross-pollinate. All right, so cross-pollination happens. Allogamy is also called cross-pollination. It only happens with flowers of the same species but a different plant. A different plant. All right, so that said, how does the process of pollination now happen? No, let's first quickly learn about the agents of pollination, because that how is what will lead us to the development of new seed. What happens at the end of the day? So agents of pollination are wind, man, water, bird, insects, okay, insect, and and man, okay. So wind pollinated flower are called the word anemone. Anemone means wind, okay. So anemophily or anemophilous flower. Anemophily is the condition of pollination by wind. Then water, hydro, right? So that will be hydrophily or hydrophilous plant or flower that is pollinated by water. Bird, the word ornithos. So ornithophily is the word the flower that is pollinated by bird. Insect, entomophily or something. Is it entophily or insect is I think it's entomophily. No, in the morph means form of insect, so it's entophily. Okay, that is insect pollinated. Then man pollinated flower. Welcome. Man pollinated flower. I think that would be like human is anthropo. Okay, just know that these are the agents of pollination. These are the agents of pollination. Now, how does pollination happen? How does pollination happen? Now, let's look at the process of pollination and how it actually takes place. The process of pollination and how it takes place. So, there's going to be a transfer of pollen grain from the anther of one flower to the stigma of another or same flower. So, now, I'm going to enlarge the structure of that male reproductive structure of the flower. Okay? All right. So this is pretty much our ovary, the ovary. All right. So now listen. So this is the stigma, remember? Stigma. And this is the style bearing the stigma. Then this is the ovarian wall, the ovary wall. Now, what happens is this: once the pollen grain gets transferred here, pollen grains are going to drop on this place. Okay, not one, not two, they are going to be many sometimes. Because, for example, an insect will just, by trying to get nectar from the flower, will be rubbing on this stigma, and the pollens from that its feather took, or that got stuck on its feather, the pollens that it took from there is going to bring it to another flower, and those pollens have characteristics depending on, for example, an insect pollinated flower has to be brightly colored, large petals, but a water pollinated flower does not have to have brightly colored petals or white petals. Do you understand? A wind pollinated flower needs to have pollens that are that are light. Okay, they are very lightweighted so that they can be carried by water. Okay, so the pollen get here. When the pollen gets here, now a pollen looks something like this. Now there is this side is called the intine. The inner portion here is called the intine, and this one outside here. Now there is, sorry, yes, intine, and this one is the exine. This one here is the exine. Alright, so this place is the intine, this place is the exine, this is the exine, the outer wall. This is a pollen grain I'm drawing for you now so that you understand. One pollen grain, this is the way it looks. Now there are two cells inside the pollen grain. Actually, initially, before the transfer, it is one cell that is there. It is once it is transferred that the cell divides mitotically. It divides by mitosis into two cells. And by the time it divides by mitosis into two cells, what will happen is that it's going to generate two cells, which we call the generative cell. We call them the generative cell and the tube cell. The tube cell. Alright, so this is the tube cell. There will be a nucleus here, there will be a nucleus here. Alright, then this pore here is the germ pore. The germ pore. Now, what happens is this. Immediately the guy gets here and the division has occurred, what will happen also is that this exine will degenerate, it will disintegrate. Once it disintegrates, the pollen grain would have doubled up here and will begin to grow. There are two cells here inside now. There are now two cells. So this guy will now begin to grow downward. Begins to grow downward. Begins to grow downward. Take note of the fact that I'm drawing more than one. Yes. So because fertilization can happen to all the ovules here. But the type of fertilization that takes place here is called double fertilization. And I will explain how. Double fertilization. We're already on development of new seed, okay? Double fertilization. So I said that this guy will now grow. Now there are two cells here already. This guy have ruptured. Is now remaining the intine. Inside this guy, there are two cells. There is the first cell here, which is going to be the generative cell, or which has the generative nucleus in it. So generative nucleus, we are particular about the content now, so we are using generative nucleus. Then there is a nucleus that is leading here. That's why this guy is able, you see that this guy is able to divert correctly, such that it will enter through this place. Do you understand now? It will enter through this place. Now, what am I saying is that the generative nucleus, grows here. Now, this one is the tube nucleus. The tube nucleus is the one that leads this pollen tube to grow. So this guy is called the pollen tube. The pollen tube begins to germinate. So let me reiterate what happens. Once there is a transfer, the exine disintegrates. This guy takes up sugar from this stigma. There's sugar on this stigma. And once it takes up the sugar and water, it swells up and begins to germinate. Do you understand? So as it begins to germinate, it grows down in this direction, being led by the tube nucleus. It enters into this place and then is able to penetrate the ovule, any of these ovules. Is that clear? Now, an ovule pretty much looks like, so you already understand the way it goes, right? Now, there's something about the tube nucleus. It is the tube nucleus that now divides by mitosis again into the male nuclei, the male nuclei. Okay, then this one now divides by meiosis, undergoes meiosis to divide into two male nuclei now. Divide into male nucleus, and this one now divides into two nuclei. Is that okay? The first male nuclei will fertilize something, the second will fertilize the other. Is that clear? Keep in mind the two nucleus divides again. Is that clear? Divides into male nuclei, and the male nuclei undergoes meiosis to divide into two cells again. Now what happens at the end of the day? Okay, also, how is it that this guy is able to know the direction it's going to? It is because it's not the tube nucleus is able to, you see, inside this oval, there are hormones called synergid hormones. Synergid hormones. I'm going to talk about it. Synergid hormone. They are the ones that help they are chemicals that help this guy to be able to be directed, the pollen tube to be directed exactly into the oval. All right. Now, how does an oval look? An oval looks pretty much like this. The point of attachment of the oval to the placenta is called the hilum. So this way is called the hilum. And the oval looks pretty much like this. This thing that attaches it is called the funicle. It's called the funicle. So now this is what we call the embryo sac. The embryo sac. And inside the embryo sac is where we now have our we have the synergids, we have the secondary nucleus, and the egg. So this is the egg cell. This is the egg cell or egg nucleus. And there is the secondary nucleus. There are two reasons. There's a reason why we have the egg cell and the secondary nucleus. The egg cell is the main thing. The secondary nucleus is for germination, for development of an endosperm. Exactly. It's for development of an endosperm, so that the endosperm, for example, in monocot, that is what the this thing considered will no, the fruit will feed on, the developing embryo will feed on. Exactly. So the secondary nucleus, all right. Of course, these layers here, the inner one is called the inner integument, and the outer one is called the outer integument. All right. Then this place here is called the micropyle. Now look at it. You see that it looks like a gate, right? The word micro means small. Pile means gate, so that's small gate. Do you understand? Now what it does is that, so this is the hilum, this is our placenta, the point of attachment of the ovule to the ovary. So this is called the funicle or the funiculus. The funicle is what holds the ovule. This is the ovule here, holds it in place. Now the point is now that that guy will now grow into this place and enter through the micropyle. Once it enters through the micropyle, the tube nucleus disappears. Then the generative nucleus, I said, sorry, it's not the tube nucleus, it's the generative nucleus, sorry, that divides into the male nuclei. I'll repeat that, sorry. The tube nucleus disappears once it enters this place. The generative nucleus is one that now divides into two nuclei, and the first nuclei will fertilize the egg nucleus. Once it fertilizes the egg nucleus, it forms the embryo, and it forms the zygote. Hey, Jesus Christ. The generative nucleus, the male nucleus, the first male nucleus, plus the egg nucleus will form the zygote. Okay, please just pardon me. I don't know why I'm trying to rush. Okay. Then the second male nucleus will now fertilize the secondary nucleus, and that's why it's called double fertilization. Okay, this one will form a diploid or diploid zygote, while this one will develop into a triploid zygote, because this guy is diploid. Okay, and the meiotic division has happened. It has given us a haploid. So n plus 2n gives us what? A 3n. That's a triploid organism. So the endosperm is triploidy. As triploidy, exhibits triploidy. All right, so that is pretty much it. This guy, and there is also a portion here that is called the nucellus. The nucellus is nutritive in function, okay, has nutritive function. Um, there are also what we call antipodal cells here. Their functions are not known, okay, antipodal cells. Their functions are not really known. So, but that's the point. So, once this happens and there's fertilization, our zygote is formed. Now, with the formed zygote, what do we have? We already have a new organism. Do you understand? Now, the new organism is basically a fruit. Now, the part, so you will now get that the part of that thing that forms so that ovule is one that becomes the seed. That ovule is one that becomes the what? The seed. While the ovary is the one that becomes the fruit. Don't forget, the fruit. And the wall of the ovary, the fruit wall. Do you understand? The seed is the oval, that oval that we just talked about. So now, that's what we're talking about now. Now, how does a new seed now develop? So a new seed typically has the following parts. It has the radicle, which is the primitive or embryonic, not primitive, primordial, the embryonic roots. That is, the roots, what will develop into the roots. So radical roots, don't forget, RR, radical develops into the roots. Then the plumule is the embryonic shoots, more like stem. Think of the shoot as stem. Is that clear? We'll call it shoot in the small plant. So embryonic shoot, okay? And then we now have cotyledons, which might be one or two. If it's one, it will be a monocot plant. If it's two, it will be a dicot plant, okay? Usually, we see the type of germination we see with dicot plant is epigeo. We are going to talk about it now. Epigeo, and with monocot, we see hypogeo germination. Hypogeo germination. So when the cotyledon is one, monocot, the cotyledon two, dicot. So for example, beans, cowpea, rather, is dicot. But you see maize, for example, is what? A monocot. And a maize pretty much looks like this. A maize seed that just developed pretty much looks like this. Now this is the radicle, okay? Yes, thank you, welcome. This is called the tegmen. This is the radicle. This is the plumule. Okay, it has an extension. That extension is the plumule. Okay. All right. Now there is this part that protects the plumule. It's called the one that protects the plumule is called the plumule sheet. Okay, the plumule sheet is called the coleoptile or coleoptile. Something like that. I think it's coleoptile. Then the one that protects the radicle is called the coleorhiza. Coleorhiza. You might see double R. It's the same spelling. It's the same thing. Protects or covers the radicle. Exactly. And this is the tegmen and this is the endosperm. This is the endosperm. Now, like I was saying, I said that it has four parts: cotyledon and the endosperm. The endosperm. Right. So these are the parts of the developing seed. Now, how does it now germinate? When you now plant that seed that has been formed, how does it germinate? There are two types of germination. We have the epigeo germination. We have the epigeo germination, and the hypogeo germination. Apogee germination. So for apogee to germinate, the tester is going to split. But here it is the pericarp that splits. The pericarp of the seed splits. Okay? In apogee germination, the hypocotyl, which turns into the shoot, the hypocotyl is first, the cotyledon is carried above the hypocotyl. The cotyledon is above the earth. That's the meaning. Epigeal is from the word epigeos, which means the cotyledon is carried above the earth. Okay? Above the earth. Okay? Suspended above the earth. That is, comes out of the ground. But hypogeal, the epigeal, so it means that here the cotyledon is above the hypocotyl. So cotyledon is also above the hypocotyl. Hypo, that is hypocotyl. Okay? The cotyledon is above it. But with hypogeal, the hypocotyl is above the cotyledon. Why? Because the cotyledon remains inside the ground. Cotyledon remains in the ground. Do you understand? Right? So that's why a maize, no, I said that monocots are the ones that we usually see show apogee germination, that's monocot, and epigeo germination is seen in dicot. So that's pretty much it. Then remember, the radicle becomes the root and the pluma becomes the shoot. God bless you. As do as subscribe to the channel. I hope I have done justice to the topic a little. Make sure you click the bell notification and like the video. God bless you.